To understand the living world today, we must journey back across deep geological time to witness one of evolution's greatest triumphs. Hi there. We're introducing a new feature where I can read this aloud in a natural, human-like voice. From frozen mountain slopes to scorching desert sands, over 15,000 described species thrive in virtually every terrestrial habitat. Yet, their story began far before human civilization, before mammals walked upright, even before the fall of the dinosaurs. For over 140 million years, ants have been continuously refining their biological and social machinery. Frozen in tree resin from the Cretaceous period, fossilized ants provide a direct window into their evolutionary origin story. They transitioned from solitary ground-dwelling hunters into complex superorganisms capable of conquering the biosphere. Their evolution is a story of cooperation overcoming individual limitation, where collective intelligence reshaped nature. Their evolution is a story of cooperation overcoming individual limitation, where collective intelligence reshaped nature. They became the prime soil tillers of Earth, aerating land and cycling organic nutrients long before earthworms evolved. How did a humble group of predatory wasps transform into the ultimate terrestrial empire? The answer lies in deep evolutionary history. Taxonomically, ants belong to the order Hymenoptera, placing them in the same evolutionary lineage as bees and wasps. To trace their roots, we must examine their closest living relatives, solitary aculeate wasps of the early Cretaceous. Over tens of millions of years, ancestral wasps surrendered the freedom of flight to claim the untamed world beneath the leaf litter. In the shadow of giant sauropods, these early pioneers laid the foundation for the most successful social experiment in nature. Their eyes witnessed the shifting of continents, the birth of flowering plants, and the rise and fall of entire animal dynasties. Physical adaptations like the flexible petiole waist and specialized chemical glands marked the birth of the true ant lineage. Paleontologists around the globe meticulously analyze these amber time capsules to reconstruct ancient evolutionary trees. As continental drift split ancient landmasses apart, early ant populations diversified across isolated geographic regions. Living fossils alive today offer vital clues, showing us primitive traits that bridge the anatomical gap between wasps and ants. The venomous sting, inherited from wasp ancestors, was repurposed from a prey-subduing weapon into a chemical defense tool. Coevolution with fungi, plants, and microorganisms pushed ant evolutionary innovation into overdrive. This is the epic chronicle of how ground-dwelling scavengers became the dominant masters of planet Earth. Welcome to the evolution of ants, a fascinating story, a deep dive into the microscopic empires shaping our world. The story of ants is inextricably bound to the transformation of soil ecology across geological epochs. Biomechanical refinements allowed early ants to master locomotion across challenging subterranean terrains. Underground refugia shielded early colonies from surface climate volatility, granting exceptional survival capabilities. Subterranean brood protection minimized egg mortality, enabling steady population growth across generations. The petiole node granted unprecedented gaster flexibility, allowing ants to sting and spray in tight soil spaces. Living underground required abandoning visual reliance in favor of tactile and chemical communication systems. Antennal sensitivity evolved to detect single molecules of pheromone signals amidst ambient environmental noise. Constantly cleaning sensory organs ensured that chemical receptors remained hypersensitive to incoming signals. This sensory mastery turned the subterranean darkness into an information-rich communication highway. In the early days of ant evolution, colonies were small, often numbering fewer than 50 individuals. Early larvae were fed directly with dismembered insect prey, a habit retained from solitary hunting wasps. Cooperative nest founding reduced individual mortality, laying the groundwork for true sociality. Territorial defense behaviors emerged early, protecting shared offspring from competing soil predators. As colony sizes expanded, subterranean architecture evolved to regulate internal humidity and temperature. I reduction occurred in ground foraging workers, reallocating metabolic energy toward olfactory neural processing. Mating flights allowed genetic mixing between isolated colonies, driving adaptive diversity across populations. Wing shedding after mating signaled the transition from aerial disperser to subterranean colony founder. Absorbed flight muscles provided vital internal nutrients, enabling founding queens to raise first broods in isolation. The birth of the first worker marked the transformation from a single female into a functional collective. Evolutionary selection experimented with diverse founding strategies to overcome high early mortality rates. Solitary foraging was the ancestral norm, preceding the evolution of mass recruitment and scent trails. In these Mesozoic leaf litters, early ants carved out specialized niches as opportunistic predators and scavengers. Though humble in their origins, these Mesozoic pioneers carried the genetic code for global dominance. Their stage was set, their body plan established. Now began the remarkable story of their diversification. During the early Cretaceous period, roughly 140 million years ago, ant ancestors were solitary wasps. These ancestral wasps hunted small invertebrates, dragging prey into primitive ground burrows for their larvae. Natural selection favored individuals with increased abdominal flexibility, granting greater agility in narrow burrows. Flight required significant energy. For ground foraging hunters, shedding wings permanently offered a massive metabolic advantage. As these wasps spent more time beneath soil debris, natural selection favored streamlined bodies and shortened legs. Mouthparts evolved from simple liquid sipping tubes into versatile mandibles capable of excavating dense clay soil. Unlike solitary wasps that provisioned nests once, early ant ancestors began feeding larvae continuously. Glandular evolution permitted primitive wasps to secrete rudimentary chemical signals, marking the dawn of pheromone language. The Mesozoic forest floor was a brutal environment, filled with predatory beetles, centipedes, and early arachnids. Venom chemistry evolved alongside physical agility, allowing early ancestors to overpower dangerous prey with surgical precision. Individual tolerance grew. Ancestral females began sharing nesting sites, reducing the high cost of solitary construction. Phylogenomic analysis confirms that ants share a deep common ancestor with apoid wasps and apoidea bees. Trees secreted thick, aromatic resin to heal bark wounds, unintentionally creating sticky traps for foraging Cretaceous insects. Over millions of years, volatile compounds evaporated from the resin, transforming it into durable, golden amber fossils. Burmese amber from Myanmar, dated to 99 million years ago, provides an unprecedented record of early ant evolution. These transitional fossils reveal mosaic evolution, organisms displaying a mixture of ancestral wasp and derived ant traits. Taxonomists place these early evolutionary bridges into the extinct subfamily Sphecomyrmenae, the wasp ants. Discovered in New Jersey amber in 1966, Sphecomyrma freyi provided concrete proof of the wasp-to-ant transition. Sphecomyrma possessed two-toothed wasp mandibles, yet featured a distinctly ant-like petiole waist and metapleural gland. The emergence of the metapleural gland was an evolutionary milestone, producing potent antimicrobial compounds for soil living. This chemical gland allowed early ants to survive in soil packed with deadly bacteria and pathogenic fungi. Brood care became increasingly specialized, laying the behavioral groundwork for true social structure. Evolution split female body plans into two distinct paths: winged reproductive queens and wingless, hard-working theriomorphs. This morphological dimorphism enabled unprecedented specialization, maximizing reproductive output while optimizing foraging. Though primitive ants were rare during the Cretaceous, accounting for less than 1% of insects, They were poised for explosion. Geological instabilities and changing climates pushed these early soil dwellers to adapt or perish. Underground competition forced early ants to refine their armor, jaw mechanics, and chemical arsenal. Early nests were modest burrows with few chambers, housing small colonies of merely dozens of individuals. Dietary shifts from purely carnivorous hunting toward opportunistic sugar scavenging provided new energetic pathways. Elongated antennal scapes evolved, granting greater flexibility to probe dark tunnels using tactile and chemical senses. The reproductive strategy of nuptial flights, followed by self-wing removal, evolved early in the Formicidae lineage. Visual systems adapted to low-light soil environments, reducing compound eye size while enhancing olfactory sensitivity. Sanitation behaviors evolved rapidly in subterranean conditions, minimizing disease transmission within crowded spaces. Behavioral thermoregulation allowed primitive colonies to maintain stable internal microclimates despite environmental swings. Global amber deposits demonstrate that early ants had spread across both northern and southern landmasses by 90 million years ago. Among the strangest Cretaceous lineages were the Hydomyrmecinae, or hell ants, equipped with vertical trap jaws. Hell ants used upward-moving mandibles and head horns to pin prey against their forehead, an evolutionary experiment now extinct. Some Cretaceous species incorporated heavy metals like zinc and iron into jaw tips for extra piercing power. Zygrasemessia, another extinct Cretaceous genus, featured dense rows of mandibular spines designed for armored prey. These early lineages represent evolutionary experiments. Most perished during the end Cretaceous mass extinction event. Extant primitive ants, such as ponerines and bulldog ants, provide living models of these ancient evolutionary stages. Australia's bulldog ants retain large eyes, solitary foraging habits, and painful stings, hallmarks of ancestral ants. In primitive species, worker cooperation remains rudimentary, with individuals foraging alone rather than in recruited lines. As evolution progressed, some lineages added a second waist segment, the postpetiole, granting supreme abdominal agility. Soil excavation capabilities unlocked new ecological niches, protecting colonies from surface heat and predators. By the late Cretaceous, the stage was set for a dramatic ecological transformation that would change the planet forever. When a massive asteroid struck Earth 66 million years ago, dinosaurs vanished, but subterranean ants endured. Their underground lifestyle and omnivorous diets shielded ants from the global catastrophic fires and impact winter. Emerging from the K-Peak extinction, ants stood ready to co-evolve with a revolutionary new group of organisms, flowering plants. Conflict over resources drove the evolution of chemical warfare and physical caste specialization. The metapleural gland's chemical secretions acted as a powerful broad-spectrum antibiotic shield. Plant cavities offered ready-made shelter, initiating a long evolutionary partnership between ants and flora. Neural rewiring accompanied social evolution, enlarging brain regions responsible for sensory integration and learning. Waste management behaviors evolved early to prevent tunnel blockages and fungal pathogen outbreaks. Plant secretions provided high-energy sugar, reducing reliance on risky live prey hunting. Controlled aerial gliding evolved in canopy species, allowing fallen workers to return safely to tree trunks. Warm Mesozoic climates permitted ants to spread into high-latitude regions across Greenland and Antarctica. Chiromyrma from 92-million-year-old amber represents one of the earliest definitive ancestors of modern subfamilies. The degradation of the venom stinger into a spraying nozzle marked a major evolutionary transition in defensive strategy. Seasonal cold tolerance mechanisms began emerging in high-latitude Cretaceous ant populations. Accidental pollen transport on ant bodies initiated early, albeit inefficient, plant pollination interactions. Prey specialization emerged early, with certain Cretaceous ant lineages hunting specific soil invertebrates like termites. Claustral founding, where the queen raises her first brood entirely on internal reserves, became a dominant strategy. Dracula ants retain non-lethal larval feeding habits, offering a living window into primitive colony nutrition. Linear serrated jaws allowed primitive hunters to pin fast-moving soil invertebrates in narrow earth crevices. Subterranean flood survival behaviors evolved, strengthening collective emergency response protocols. From humble solitary origins, the ant body plan was perfected, poised to inherit the post-dinosaur world. The Paleogene era witnessed the rapid diversification of angiosperms flowering plants that reshaped terrestrial biomes. Flowering plants evolved extrafloral nectaries, specialized glands offering sugary nectar solely to recruit ant bodyguards. In exchange for nectar, ants aggressively cleared plants of herbivorous insects, establishing a powerful mutualism. Trees evolved hollow structures like domatia, offering built-in housing designed specifically to shelter ant colonies. Pea plants synthe-sized lipiad and protein reach FOOD angiosperms also developed myrmecochory, seed dispersal by ants, utilizing nutrient-rich seed appendages called elaiosomes. This subterranean planting protected seeds from seed-eating birds and bushfires, boosting plant survival rates dramatically. Though less efficient pollinators than bees, some ant species contributed to cross-pollination in low-canopy ecosystems. Genetic dating reveals that major ant lineages diversified in tandem with the expansion of tropical rainforest canopies. Ants ascended from the dark forest floor into the leafy canopy, occupying arboreal niches previously untouched by insects. Arboreal life demanded novel engineering. Weaver ants link their bodies together to pull heavy leaves into tight enclosures. Using their own larvae as living glue guns, weaver ants stitch leaves together with silk to craft high canopy nests. Canopy nesting protected colonies from ground floods and soil predators, unlocking vast new vertical territories. Ants domesticated these hemipterans, herding them like livestock and harvesting their carbohydrate-rich honeydew. Ant bodyguards fiercely defend their aphid herds against ladybugs, lacewings, and parasitic wasps day and night. When plants withered, worker ants physically transported their insect livestock to fresh, healthy foliage. Underground, ants farmed root-feeding aphids, securing reliable sugar supplies even through harsh seasons. Some species construct protective barns made of carton or soil over their sap-sucking herds to shelter them from weather. Honeydew provided massive caloric energy, fueling rapid colony growth and allowing ant populations to explode. Eocene Baltic amber, dated to 40 million years ago, demonstrates that ants had become ecologically dominant insects. By the Eocene epoch, modern ant genera like Lasius, Formica, and Camponotus were already well established. Fossilized leaves from Wyoming reveal leaf-biting behaviors identical to those seen in modern carpenter ant nests. In less than 40 million years, ant abundance rose from under 1% to over 10% of all fossil insects. Niche partitioning evolved. Different species specialized in different canopy heights, times of day, and prey sizes. Night foraging species developed enhanced light-gathering eyes and specialized optical neural pathways for dark navigation. Miniaturization allowed small ant species to utilize microcavities inside seeds, stems, and soil crevices. In contrast, giant extinct species like Titanomyrma reached wingspans over 25 centimeters during warm Eocene climates. As global temperatures dropped during the Oligocene, ants adapted to seasonal cold, expanding into temperate zones. Temperate ants evolved diapause, a physiological winter dormancy akin to hibernation to survive freezing winters. Cold-tolerant ants produced natural antifreeze molecules in their blood, preventing icy crystal formation in cells. As spring sun warmed surface mounds, workers acted as living solar heaters, carrying warmth back down to brood rooms. In arid grasslands, harvester ants evolved, utilizing tough mandibles to mill dry plant seeds into nutritious ant bread. Specialized soldier castes with enlarged heads evolved specifically to crush tough seed husks for the worker force. Glandular secretions applied to stored seeds inhibited mold growth, creating long-term strategic food reserves. Seed harvesting allowed ant colonies to thrive in dry, seasonal environments where insect prey was scarce. Desert species evolved incredible navigational abilities, utilizing polarized skylight patterns and step-counting path integration. By calculating directional vectors and distance traveled, desert ants returned to subterranean burrows in straight lines. Morphological shifts like elongated legs raised body heights above blistering sand surface temperatures. High-speed tripod gaits developed, minimizing foot contact time with scorching desert sand surfaces. Reflective hair structures evolved to reject solar radiation and emit internal heat back into the atmosphere. Water collection became an essential colony duty in arid zones, with workers storing liquid internally to hydrate nestmates. In North American and Australian deserts, honeypot ants evolved living storage vessels to stockpile liquid reserves. These living storage workers, called repletes, expand their abdomens dramatically to store nectar for dry seasons. During severe droughts, the entire colony relies on honeypot repletes for life-sustaining water and carbohydrates. Highly stretchable chitinous membranes allowed the gaster to expand to many times its normal resting volume. The mound architecture evolved aerodynamic shapes, preventing wind erosion while maximizing interior ventilation. Through coevolution with plants and extreme physiological adaptations, ants conquered every terrestrial frontier. Their adaptability across changing climates and landscapes was made possible by one ultimate secret: eusociality. To fully grasp their evolutionary triumph, we must examine the origin and mechanics of the superorganism. Plant galls provided additional carbohydrate sources, strengthening ant presence in temperate forest canopies. Neotropical Cecropia trees evolved specialized food bodies called Müllerian bodies to feed resident Asisteca ants. Müllerian bodies, rich in glycogen, represent a rare instance of plants synthesizing animal-like glycogen for ant mutualists. The ants efficiently funneled plant carbohydrates into collective biomass, outcompeting solitary plant-eating insects. Carton nest construction allowed arboreal species to expand, independent of natural tree hollow availability. Durable salivary glues rendered carton nests waterproof, enduring tropical monsoons without structural damage. In North American deciduous forests, spring ephemeral wildflower dispersal relies almost entirely on myrmecochory. Subterranean waste pits offered ideal microenvironments: moist, nutrient-rich soil protected from seed predators. This plant-ant partnership shaped spring woodland flora, driving plant diversity across temperate continents. Coniferous forests offered honeydew resources through aphids, supporting massive wood ant mound networks in Europe. Wood ant colonies collect hundreds of kilograms of honeydew annually, fueling massive energy demands. Internal crop storage permitted efficient carbohydrate transport back to subterranean nurses and larvae. The crop acted as a shared energy reservoir, sustaining the collective workforce through intensive foraging trips. Clearing surrounding plants reduced fire risk and eliminated cover for predatory spiders near the nest entrance. Mandibular mechanics evolved flat crushing surfaces, perfect for grinding tough grain starches into edible paste. Coevolution with angiosperms transformed ants from simple ground scavengers into dominant ecological engineers. Eusociality, the highest level of social organization, is defined by three traits: reproductive division of labor, overlapping generations. In a eusocial colony, individual self-interest is sacrificed for collective reproductive success. A key driver of hymenopteran social evolution was haplodiploidy. Males develop from unfertilized haploid eggs. Because full sisters share 3/4 of their genes, evolutionary kin selection made helping raise sisters genetically advantageous. Workers pass on their genetic heritage indirectly by ensuring the survival of their fertile sisters and queen. Haploid males, or drones, exist solely to carry genetic material to new queens during nuptial mating flights. During nuptial flights, young queens and males launch into the sky, mixing genetic lineages across broad landscapes. Once mated, a queen sheds her wings permanently, storing millions of sperm inside a specialized organ called the spermatheca. Sperm stored within the spermatheca remains viable for up to 30 years, fertilizing millions of eggs without remating. The claustral founding process is extremely dangerous. A young queen seals herself in total darkness to raise her first brood. Without foraging outside, the queen metabolizes her useless flight muscles to produce protein-rich saliva for her larvae. When the first miniature workers emerge, they take over nest excavation and foraging, freeing the queen to focus solely on egg production. From a single vulnerable queen, colony populations grow exponentially, sometimes reaching millions of workers over decades. Task allocation in the colony is governed by temporal polyethism. Workers change jobs as they grow older. Young, soft-bodied workers remain deep inside the nest, serving as nurses for eggs, larvae, and the queen. As workers age, they transition outward, taking on nest construction, tunnel maintenance, and sanitation duties. The oldest workers undertake the most dangerous assignment: foraging in the outside world filled with predators. The colony is governed by chemical language. Ants leave faint pheromone trails to mark paths toward rich food sources. Upon discovering food, a worker deposits recruitment pheromones, creating an invisible chemical highway for nestmates. Positive feedback loops emerge. As more ants follow the trail and reinforce the scent, recruitment accelerates rapidly. Antennal tapping, or antennation, transfers tactile and chemical signals regarding hunger, threat levels, and caste identity. Every colony possesses a unique chemical signature, composed of cuticular hydrocarbons on the exoskeleton surface. By sweeping antennae across arriving workers, nest guards instantly identify nestmates or detect foreign intruders. The queen secretes powerful inhibitory pheromones that suppress ovary development in workers and signal colony health. As workers share food mouth to mouth, queen pheromones propagate through the colony, maintaining social cohesion. When an ant dies, its body releases fatty acids like oleic acid, signaling workers to transport the corpse to a refuse pit. Necrophoric behavior prevents lethal pathogens from spreading from decomposing bodies into sensitive nursery chambers. Dedicated sanitation workers manage waste pits, sacrificing their own health to shield the colony from disease. Individual hygiene is meticulous. Ants spend up to 8 hours a day grooming themselves and their nestmates. Antimicrobial compounds secreted by body glands turn the ant exoskeleton into a self-sterilizing surface. In a superorganism, individual ants act like cells in a body. Workers are organs, the queen is the ovary, and chemical trails are nerves. This distributed intelligence allows the collective to solve complex environmental challenges far beyond individual capacity. Without a central leader or mastermind, local interactions between individual ants produce global structural solutions. Living bridges self-assemble and adjust dynamically, optimizing traffic speed while minimizing the number of ants trapped in the structure. Nomadic species build living nests or bivouacs, using interlocked claws and legs to protect the queen and brood without soil. Interlocking leg claws form strong, flexible biological fabrics capable of enduring severe wind, rain, and physical impact. Superorganisms maintain homeostasis, regulating temperature, humidity, oxygen, and carbon dioxide within narrow limits. Workers adjust tunnel entrances based on atmospheric cues, creating natural air conditioning systems throughout the nest. Humidity regulation prevents eggs and larvae from desiccation, ensuring high reproductive success rates. Morphological caste differentiation produces specialized physiological tools optimized for specific tasks. In polymorphic species like Pheidole, major workers possess heads 4 times larger than minor workers, packed with jaw muscles. Major workers act as biological nutcrackers, processing hard food items that minor workers bring back to the nest. Enormous head capsules accommodate massive muscle bundles, generating extreme crushing force at mandible tips. Nutrient processing is fully integrated. Major workers crush prey, minor workers digest it, and nurses feed the larvae. Adult ants cannot digest solid food. They feed solid proteins to larvae, which digest them and regurgitate nutrient fluids back. This metabolic loop makes larvae essential internal organs of digestion for the entire superorganism. Trophallaxis ensures that food distribution is equitable, keeping every caste nourished regardless of individual foraging success. Ants exhibit rescue behaviors, dragging wounded nestmates out of danger and nursing them back to health inside the nest. Recent scientific studies confirm that ants perform medical treatments, applying natural antibiotics to infected wounds. The evolutionary emergence of the superorganism converted fragile individual insects into formidable ecological powerhouses. This social architecture provided the evolutionary springboard for unprecedented ecological diversification across the globe. Worker policing prevents unauthorized worker reproduction, maintaining social harmony and queen reproductive dominance. Workers detect non-queen egg odors. Unauthorized eggs and reproductive workers must be punished. Epigenetic switches modulate gene expression, producing distinct brains and behavioral suites from identical genomes. Dynamic brood movement optimizes embryonic development rates in response to shifting soil microclimates. Foragers act as collective sensors, adjusting foraging effort based on internal colony hunger levels. The proventriculus valve prevents crop contents from entering the worker's stomach, preserving food for sharing. Positive feedback loops allow superorganisms to rapidly exploit large food discoveries before competitors arrive. Collective intelligence computes geometric shortcuts without any single ant possessing a global map of the space. Distributed routing algorithms allow ant superorganisms to bypass network disruptions with extreme resilience. Ritualized displays prevent costly physical combat when territorial boundaries can be settled by numbers alone. Substrate-borne acoustic communication alerts distant nestmates to cave-ins and predator attacks instantaneously. Collective panic protocols ensure orderly evacuation during life-threatening environmental disasters. Superorganismic organization drastically lowers metabolic maintenance costs per individual, boosting survival efficiency. Age-dependent risk allocation dispatches older workers with lower remaining lifespans to high-mortality frontlines. Individual sacrifice, chemical language, and collective intelligence forged the ultimate social machine. In the superorganism, the collective becomes the individual, achieving ecological immortality through continuous renewal. This unmatched social synergy allowed ants to radiate into specialized subfamilies across every continent. As ants spread across continents, evolution branched into distinct subfamilies, each pioneering unique survival strategies. Ponerines rely on potent stings and mechanical power, hunting single prey items in dark forest leaf litter. In India, Harpagnathos saltator evolved elongated mandibles and jumping capabilities to ambush agile prey. When a Ponerine queen dies, workers engage in ritual duels to become gamergates, reproductively active workers that replace the queen. This neurohormonal transformation extends worker lifespans from months to years, preserving the colony's survival. The subfamily Myrmicinae became the most diverse ant group on Earth, boasting over 6,000 specialized species. Within Myrmicinae, leafcutter ants evolved fungus farming, an agricultural innovation that transformed tropical ecology. Colonies construct cleared forest highways stretching up to 100 meters, moving tons of vegetation underground. Deep underground, workers process leaves into pulp to feed a specialized cultivar fungus, Loco agaricus gondylophorus. The fungal garden produces gondylidia, nutrient-rich structures grown specifically to feed the ant population. Miners weed the garden continuously, using mouthparts to pick out unwanted fungal spores and maintain a pure crop. Leafcutters carry symbiotic bacteria on their exoskeletons, producing natural antibiotics that kill garden-destroying Escovopsia mold. This three-way mutualism between insect, fungus, and bacterium represents one of evolution's most complex interspecies alliances. Toxic waste is deposited into deep subterranean shafts, managed by old workers who spend their remaining lives isolated in waste pits. The subfamily Formicinae abandoned venomous stings entirely, developing long-range chemical artillery instead. Formicines possess an acidopore, a specialized circular nozzle that shoots concentrated formic acid at enemies. Wood ant mounds deploy synchronized chemical defense, spraying cloudbursts of formic acid to repel bears and birds. Formic acid concentration in their venom glands reaches up to 6%, strong enough to blind large vertebrate predators. Formicinae also includes carpenter ants, counts, master wood excavators that hollow out dead trees for nest sites. Carpenter ants do not eat wood. They excavate intricate gallery networks within timber to shelter growing colonies. In arid regions, Formicine honeypot ants evolved living storage tanks to preserve liquid carbohydrates through dry spells. The subfamily Dolichoderinae evolved volatile chemical weapons, producing pungent defensive sprays from anal glands. Dolichoderines spray defensive compounds like iridomyrmecin, which disrupt the nervous systems of competing ant species. Argentine ants form global supercolonies, utilizing low genetic diversity to merge nests across thousands of kilometers. The subfamily Dorylinae comprises army ants and driver ants, nomadic predators that abandon permanent nests entirely. Army ant soldiers feature giant curved mandibles designed exclusively to pierce and pin prey during fast-moving raids. Over 200,000 army ants march in unified columns, consuming scorpions, tarantulas, and small reptiles. When local prey is depleted, army ant colonies dismantle their living bivouac nests and march across the forest floor. Doryline queens are dichthyodyines, wingless, massive egg factories capable of producing millions of eggs per month. In Africa, driver ant columns contain up to 20 million workers, forming the largest single-colony swarms on Earth. Evolution pushed mechanical design to absolute physical limits in trapjaw ants. Ultra-sensitive trigger hairs inside the mouth detect prey contact, firing an instantaneous neurological reflex signal. Trapjaw mandibles strike at speeds over 200 kilometers per hour, generating forces 300 times the ant's body weight. Latch-mediated spring actuators store elastic energy in head chitin, releasing it instantaneously like a loaded crossbow. When threatened, trapjaw ants strike the ground, using recoil energy to launch themselves backward into safety. In the rainforests of Borneo, Attocalabopsis explodens evolved autothesis, sacrificial self-detonation in defense. Worker ants rupture their body walls, releasing sticky, toxic glandular goo that immobilizes and kills intruders. Boreal turtle ants evolved phragmosis, specialized saucer-shaped heads used as living corks to block tunnel entrances. Their armored headplates fit tree cavities perfectly, creating impenetrable physical barriers against invading species. Sahara silver ants evolved heat shock proteins and optical physical coatings to forage in extreme midday heat. Triangular hairs reflect visible and near-infrared light while radiating body heat, keeping internal temperatures below lethal limits. The bullet ant possesses a sting rated as the most agonizing in the entire insect world. Its venom contains panerotoxin, a neurotoxin that blocks sodium channels, causing burning pain that lasts 24 hours. Weaver ants build complex aerial leaf tents, using cooperative physical tension to bind living leaves into shelters. When leaves are far apart, workers form living chains, combining physical strength to bend heavy branches together. As ants evolved, dangerous pathogens evolved alongside them, including mind-controlling zombie fungi. The fungus forces the ant to climb exactly 25 centimeters above ground and clamp its jaws into a leaf vein. Ants evolved social immunity, behavioral defenses like quarantine and grooming to stop epidemic outbreaks. Glandular antibiotics applied during grooming neutralize fungal spores before they penetrate the outer chitin armor. This eternal evolutionary arms race continuously refines both ant behavioral defenses and pathogen infection strategies. Social parasitism emerged in several ant lineages, where parasitic species exploit the labor of host colonies. The Pacac polyergus queen eliminates the host, assimilating her pheromones. Slave-making ants raid neighboring host nests, stealing pupae to rear them as enslaved workers in their own colony. Enslaved workers perform all colony tasks, feeding host adults whose saber-shaped mandibles are useless for foraging. Ant dominance created visual niches. Hundreds of spiders, beetles, and bugs evolved body shapes mimicking ants. Myrmecomorphy, ant mimicry, allows predators to evade birds and trick ants into allowing them near their nests. Parasitic beetles secrete addictive chemical liquids that pacify worker ants, granting the beetles free rein inside the nest. Ant nests became ecological hubs, supporting thousands of parasitic, mutualistic, and commensal species. From extreme anatomical weapons to intricate symbioses, subfamily diversification cemented the ant's global supremacy. Every specialized trait evolved over millions of years, contributed to an indestructible global web of ant dominance. Non-lethal larval cannibalism in Dracula ants highlights ancestral dietary transitions preserved in deep subterranean lineages. Arboreal myrmecines evolved aerial agility and trapjaw mechanisms independently, converging on predatory precision. Mound-building fungus farmers optimized nest thermoregulation in subtropical grasslands across South America. Canopy-bound colony founding bypassed ground predators, unlocking vast vertical space in tropical forests. Weaver ants combine physical bite force with chemical spraying, driving birds and primates away from host trees. Panerotoxin synthesis in bullet ants represents a highly specialized neurotoxic adaptation against mammalian predators. Thermal stilting lowers body temperature by 5 degrees Celsius, granting vital extra minutes of desert foraging time. Dynamic body bridges allow army ant columns to maintain high marching speed across rugged terrain. Driver ant subterranean excavations alter soil density across vast African savanna ecosystems. Directed aerial gliding allows wingless canopy workers to steer toward home tree trunks with 80% accuracy. Volatile monoterpenes create pungent chemical smoke screens, disorienting larger invertebrate predators. Fungal hyphae take direct physical control of worker limb muscles, bypassing brain control during zombie manipulation. Subfamily diversification transformed ants into versatile ecological masters across every terrestrial biome. In the modern era, human global trade unwittingly unleashed ant species across ocean barriers into new continents. Invasive ants hitchhiked aboard trade ships, invading foreign ecosystems and displacing native insect species. In California, South Europe, and Japan, Argentine ants formed a single global supercolony spanning thousands of miles. Due to genetic bottlenecks during transport, invasive ants share identical colony scents, eliminating internest warfare. Red imported fire ants invaded North America, building high-density mounds and attacking native fauna. Fire ant venom contains solenopsin, an alkaloid toxin that causes burning sting pustules and severe allergic reactions. On Christmas Island, introduced yellow crazy ants swarmed native red crabs, spraying formic acid into their eyes. Yellow crazy ants devastated red crab populations, altering island forest structures and crashing local biodiversity. Little fire ants, Wasmannia oropunctata, invade tropical plantations, disrupting agriculture and blinding domestic animals. Humans developed targeted chemical baits, exploiting trophallaxis to transport slow-acting toxins directly to invasive queens. Biological control strategies introduce natural predators like phorid flies, which lay eggs inside live ant heads. Phorid fly larvae consume ant brain tissue, decapitating workers and suppressing invasive colony populations naturally. Scientific innovations like liquid metal nest casting reveal the intricate geometry and scale of subterranean architecture. Researchers attach microscopic RFID tags to individual ants, tracking collective decision-making networks in high resolution. Ant colony optimization algorithms inspire modern human technology, streamlining internet routing, AI, and delivery logistics. Roboticists design autonomous swarm micro-robots based on its simple rules to execute complex disaster rescue operations. Biomechanical engineers study ant leg joint nanostructures to design wear-resistant robotic limbs and flexible materials.
The metapleural secretions are highly effective.
Yeah.
This could be the breakthrough we needed against superbugs. Habitat loss and global climate change threaten thousands of native ant species that perform vital ecological services. As native ant species disappear, soil aeration, seed dispersal, and natural pest control networks suffer global declines. Yet, resilient species adapt rapidly to urban concrete jungles, thriving inside human buildings and thermal pollution zones. Urban ants shifted diets toward human food waste, utilizing urban garbage as high-calorie fueling stations. They adapt to human technological environments, nesting inside microelectronics and urban infrastructural conduits. As we look ahead into deep geological time, one truth remains clear. Ants will endure long after human civilization. Their genetic flexibility and social resilience ensure that ants will continue evolving, colonizing future terrestrial biomes. Subterranean protection, chemical language, and collective intelligence form an evolutionary formula resistant to mass extinction. Every new queen that burrows into soil carries forward an unbroken line of lineage stretching back to the Cretaceous era. Generation after generation, the superorganism renews itself, executing timeless behaviors written into chitin and DNA. Their silent chemical conversations will continue to echo across forest floors, deserts, and canopy branches for eons. In the vast cosmic story of life on Earth, the humble ant stands as an unmatched masterpiece of evolutionary design. They remind us that true power lies not in individual strength, but in the harmonious cooperation of the collective whole. As silent stewards of the soil, ants remain the foundational pillars sustaining terrestrial life across our planet. Their evolutionary epic is far from over. It is an ongoing story of adaptation, survival, and ecological dominance. Ants, the undisputed masters of deep time, biological engineering, and collective survival on Earth. Thank you for exploring the fascinating evolutionary story of ants. Subscribe to discover more microscopic wonders of natural science. Modern genomic sequencing reveals that the ant master genome contains highly conserved gene clusters dedicated to olfactory receptors. Transcriptomic studies show that worker caste differentiation is driven by epigenetic gene expression rather than structural DNA changes. Environmental cues and nutritional inputs trigger DNA methylation, turning specific behavioral gene networks on and off dynamically. Differential feeding alters microRNA levels in developing larvae, determining whether an embryo becomes a sterile worker or a fertile queen. Fluctuations in juvenile hormone concentrations during critical larval stages dictate final body size, mandible length, and caste fate. Polyphenism allows a single genetic code to express a continuous spectrum of worker body sizes customized for diverse ecological tasks. Gene duplication expanded ant olfactory receptor families to over 400 genes, far exceeding those found in solitary wasps. Expanded olfactory neural centers, known as antennal lobes, process dense chemical information with incredible signal resolution. Mushroom body brain structures enlarged significantly in ants, supporting spatial navigation, route learning, and social memory. Tandem running represents a true form of insect teaching. A knowledgeable worker leads a naive nestmate directly to a new food site. The Baltic region holds immense amber deposits that preserved millions of Eocene insects, capturing ant social evolution in gold. Fossilized group behaviors preserved in amber prove that complex cooperative foraging existed over 40 million years ago. Dominican amber from the Miocene epoch reveals ancient parasitic interactions identical to modern fungal infections. Ancient resin specimens document the deep evolutionary roots of ant-aphid farming relationships during the Miocene. Canadian amber from Grassy Lake provides critical evidence of early ant lineages coexisting with gymnosperm-sucking insects. French amber deposits capture transitional ant forms that survived shifting Cretaceous shorelines and floral transitions. Advanced X-ray micro-CT technology allows scientists to render 3D digital skeletons of ants hidden inside opaque amber blocks. Synchrotron radiation reveals soft tissue organs preserved inside ancient exoskeletons, revealing internal digestive evolution. Compression fossils in sedimentary rock record ancient nest gallery layouts, showing subterranean engineering through deep time. Petrified wood specimens demonstrate that arboreal wood-boring ant habits evolved early in forest ecosystem history. In the Amazon rainforest, ants move more organic matter than all mammalian herbivores combined, dominating ecosystem processing. Tropical rainforests pushed predatory army ants to refine swarm tactics, turning entire forest floors into active hunting zones. High-altitude cloud forests bred specialized cold-tolerant species capable of foraging in dense fog and low ambient heat. In Bornean peat swamps, swimming ants nest inside pitcher plants, diving into acid fluid to hunt. Specialized claw structures and waxy skin coatings allow swimming ants to navigate digestive fluids without drowning. The pitcher plant's rim is glistening with sweet nectar. The ants clean it and protect it from flies. In dry scrublands, ants construct soil shelters around plant roots, protecting delicate honeydew herds from drying winds. Honeypot ants represent an indigenous ecological food source, held in high cultural esteem by Australian First Nations peoples. Succulent specialized ants nest inside water-storing plant leaves, obtaining both humid shelter and hydration in deserts. In subarctic tundras, ants maximize short summer windows, elevating nest temperatures by building dark organic mounds. Biomechanical testing reveals that ant mandibles generate peak force pressures exceeding several thousand atmospheres. Helicoidal chitin fiber arrangements make the ant exoskeleton exceptionally resistant to fracture, impact, and bending forces. The neck joint of an ant can support forces up to 5,000 times the ant's body weight before structural failure. Rolia footpads secrete thin liquid films, utilizing capillary adhesion and van der Waals forces to walk upside down on glass. Sensory organs on leg joints, called campaniform sensilla, measure mechanical stress, allowing instant gait stability tweaks. Johnston's organ detects air movement and sound vibrations, giving ants precise acoustic perception of nearby motion. Substrate-borne vibrational signals travel through plant stems, alerting nestmates to danger faster than airborne scents. Abdominal muscle leverage allows some species to squirt chemical defenses over distances 30 times their body length. Frictional cleaning mechanisms ensure that foot adhesive pads remain free of microscopic dust particles. Capillary action and muscular pharyngeal pumps allow ants to lap up liquid carbohydrates at high volumetric speeds. Cuticular hydrocarbon profiles consist of complex mixtures of alkanes, alkenes, and methyl-branched alkanes unique to each colony. Workers constantly spread cuticular lipids across their chitin, preventing lethal body dehydration in dry air. Mandibular glands release volatile alarm compounds like 4-methyl-3-heptanone, triggering rapid defensive swarming. Trail pheromones vary in volatility. Short-term trails guide immediate recruitment, while long-term trails mark permanent paths. Pheromone evaporation dynamics naturally force ant trails to converge on the geometrically shortest path to a food source. Dufour's gland produces species-specific hydrocarbon blends used to mark territorial boundaries and recruit nestmates. Territorial marks inform rival colonies that an area is actively defended, reducing unnecessary physical conflict. Venom compositions evolve diverse chemical weapons, ranging from proteinaceous neurotoxins to cytotoxic piperidine alkaloids. As stinger mechanisms degraded in advanced lineages, venom chemistry shifted toward volatile spraying and topical toxins. Traffic rules self-organize on ant highways. Loaded returning foragers receive right-of-way priority at crowded junctions. Trail maintenance workers actively remove obstacles, fallen leaves, and debris to keep supply lines flowing smoothly. Some ground nesting species construct soil archways over trails, creating weatherproof tunnels across open terrain. Ants experience true sleep. Workers take hundreds of short power naps daily, totaling roughly 4 hours of rest. Queen ants experience longer, uninterrupted sleep cycles, contributing to their extraordinary lifespan compared to workers. Young worker ants engage in play fighting, building motor skills and testing dominance hierarchies without injury. Recruitment behaviors adapt dynamically to food quality. Higher calorie rewards trigger more enthusiastic recruitment displays. Analyzing olfactory cues, calculating spatial vectors to the nest. Parallel processing initiated. Recent behavioral experiments suggest that certain ant species show rudimentary self-awareness in mirror mark tests. Epiphytic ant plants like Andid discidia grow hollow leaves that serve as pre-made nest sites for climbing ant colonies. Ants store organic waste inside hollow ant plant leaves, providing roots with essential nitrogen in nutrient-poor rainforests. Lemon ants create devil's gardens by injecting formic acid into all non-host plant leaves. By systematically poisoning rival saplings, lemon ants eliminate plant competition, creating pure stands of their host tree. Some devil's gardens are cultivated continuously for over 800 years by successive ant generations. In Southeast Asia, albid ants cultivate fungal gardens inside living plant cavities, nourishing host stems. Nitrogen-fixing cyanobacteria cultured inside ant nests convert atmospheric nitrogen into bioavailable plant nutrients. Predatory spiders exploit ant communication, mimicking alarm vibrations to lure unsuspecting workers into ambushes. Parasitic flies hover above worker columns, striking in milliseconds to deposit eggs precisely into head capsule joints. Throughout geological time, ants turned soil excavation into an ecological art form, shaping world agriculture. In tropical and arid biomes, ants surpass earthworms as primary soil tillers, driving carbon and nutrient cycling. Ant seed dispersal planted global forests, creating resilient plant communities capable of enduring environmental shifts. As nature's premier scavengers, ants clean ecosystems, preventing pest outbreaks and disease transmission. Without ants, terrestrial ecosystems would face catastrophic soil compaction, seed stagnation, and waste accumulation. Their journey from solitary Mesozoic wasps to global superorganisms stands as one of evolution's greatest achievements. 140 million years of continuous evolution crafted a biological marvel of unmatched resilience. As long as Earth supports terrestrial life, new queens will take to the skies, founding empires deep beneath the soil. In every handful of soil, in every forest canopy, their microscopic empire endures, the true masters of time. Advanced X-ray synchrotron imaging allows scientists to reconstruct internal muscular structures of 90-million-year-old ants. Molecular communication systems evolved extreme biochemical specificity, distinguishing nestmate signatures from foreign intruders. Trophic eggs function as internal protein batteries, allowing colonies to buffer nutritional shortfalls during environmental stress. Necrophoric hygienic behaviors prevent deadly fungal pathogens from spreading through crowded subterranean brood rooms. The proventriculus valve transformed the crop into a true social stomach, enabling efficient colony-wide nutrient sharing. Mechanical energy stored in head chitin releases at extreme velocities, turning mandibular strikes into defensive catapults. Living carbohydrate storage repletes expand their abdominal membranes to survive multi-year desert droughts. Agricultural weeding behaviors maintain monoculture fungal purity, representing nature's earliest industrial farming. Bacteria carried on ant exoskeletons produce targeted antibiotics that eliminate garden-destroying Aedes aegypti mold. Obligate plant-ant mutualisms force trees to synthesize lipid-rich food bodies specifically to reward resident ant armies. Self-assembling hydrophobic rafts trap air bubbles beneath trapped workers, protecting queens and larvae during monsoons. Thermal stilting and reflective hair coatings allow desert ants to forage on sand surfaces exceeding 70 degrees Celsius. Social parasites exploit chemical communication, tricking host workers into rearing parasitic offspring. Fungal pathogens manipulate host neurochemistry, forcing ants into fatal death-grip postures on leaf veins. The superorganism acts as a single living entity, achieving ecological immortality through continuous worker replacement. Arboreal carton nests protected colonies from ground floods and soil predators, expanding vertical forest colonization. Seed milling unlocked vast carbohydrate reserves in arid grasslands, where insect prey was seasonally scarce. Myrmecomorphy allowed predators to bypass ant security and evade visual predators by mimicking ant body profiles. Myrmecophiles produce addictive chemical compounds, bribing host workers for free food and brood access. Myrmecochory planted global temperate forests, protecting plant seeds from bushfires and avian seed predators. Synchronized chemical artillery allows wood ant colonies to defend massive needle mounds against large vertebrates. Living worker chains combine physical strength to pull distant leaves into tight aerial nest enclosures. Nomadic army ant colonies construct living nests with their own interlocked bodies, abandoning permanent soil burrows. Haplodiploidy favored kin selection, driving the evolutionary transition from solitary wasps to eusocial societies. Tactile antenation transfers critical information regarding food quality, threat levels, and caste identity. Cuticular hydrocarbon profiles serve as chemical passports, allowing nest guards to identify colony members. Dynamic brood movement optimizes embryonic development rates in response to shifting soil microclimates. Nuptial flights mix genetic lineages across broad landscapes before young queens dig solitary founding burrows. Absorbed flight muscles provide vital internal nutrients, enabling founding queens to raise first broods in isolation. The birth of the first worker transforms a single founding female into a functional superorganism. Low genetic diversity in invasive populations eliminates internest aggression, creating continent-spanning supercolonies. Ant colony optimization algorithms inspire human technology, streamlining internet routing and autonomous logistics. Biomechanical nanostructures in ant leg joints inspire wear-resisting robotic limbs and flexible joint materials. Pharmacologists analyze ant chemical secretions to discover novel antibiotic drugs capable of killing superbugs. Resilient ant species adapt rapidly to urban concrete jungles, thriving inside human infrastructure. Ants integrate into human technological environments, nesting inside microelectronics and urban conduits. Subterranean protection and collective intelligence form an evolutionary formula resistant to mass extinction. In the vast cosmic story of life on Earth, the humble ant stands as an unmatched masterpiece of evolutionary design. Thank you for exploring the evolutionary story of ants. Subscribe to discover more microscopic wonders of natural science.